Hello, and welcome to today's lecture on genetically modified organisms for scientific ethics. And so what we're going to do today is review some of the concepts on recombinant DNA technology, kind of similar to what we talked about in the last lecture, where we used two pieces of DNA, combined them together with microorganisms like bacteria and yeast. So we're going to review that a little bit, talk a little bit about the kind of restrictions or the rules associated with that, and then look at more complicated organisms, kind of organisms that are higher multicellular organisms, essentially plants and animals that we can think about. So with this idea of transgenic organisms, the idea is that what we're going to be doing is modifying an organism by transferring or transferring genes into that organism. And so we're going to either insert in or delete specific genes. And so the idea is that we're going to look for kind of a desired gene, desired characteristic or trait that we're interested in. We're going to take that from one organism, put it into another organism, and in doing so, change that organism in a way that it now has characteristics or traits that we're interested in using in some way. Now, the example we've got over here on the right-hand side of the slide is a tobacco plant. And so you can see the kind of roots down here, you can see the stem growing up, and then the leaves up here. What was done with this tobacco plant is that it was inserted, or its genome was inserted with the firefly luciferase gene. So luciferase, described down here, is basically a gene that fluoresces when it's activated, or not a gene, a protein that fluoresces when it's activated. So if you've seen fireflies at night kind of flying around, you know that their kind of tail end, where they've got a lot of that luciferase being present, is going to glow at certain points. And so what we've got here is a tobacco plant that is expressing the luciferase gene, so it's going to be glowing, and you see glowing leaves, stems, and roots associated with it. And so that, again, is going to be a good example of a transgenic organism or a genetically modified organism. We've modified that organism or changed that organism by transferring genes into it. Now, as we said in the last lecture, bacteria were the first genetically modified organism. They were very, very simple, the single-celled organism, but they were capable of being genetically modified. They were going to be capable of getting these transgenes moved into them. And we talked about a couple different examples associated with it. We talked about, putting in the gene for human insulin and getting bacteria to produce the human form of insulin. And then another example, the clotting factor 8. Both of these were getting bacteria to express this human gene and produce the human protein, either insulin or clotting factor, clotting factor, that will allow it then to be less problematic. It's not going to cause an immune system response like a pig insulin. It doesn't have the infectious disease risks associated with it, like the clotting factors. But basically what we're looking at here, again, a scientific laboratory setting, a controlled setting, where we've got these stainless steel vats under controlled circumstances, controlled environments, where we're going to be growing the bacteria and then isolating whatever protein it is that these bacteria now are expressing. Now, there were a lot of concerns that we're going to go in, we're playing God, we're changing the natural evolution of things, natural kind of development of things, if we go through and recombine pieces of DNA that were never kind of naturally meant to come together with one another. So the National Institute of Health, again, we've talked about this throughout the course, the National Institute of Health is the primary supporter of biomedical research within the United States. In the early 1970s, recognized that this concept and this technology of recombining DNA was going to be kind of an emerging field. And they established a recombinant DNA advisory committee in 1974. And this committee continues to work, has come up with lots of rules and regulations for the appropriate biosafety practices and containment measures to be working with genetically modified organism. Again, because you don't want to, kind of be creating something and then have the risk that it's going to cause, get it released in the environment, causing problems, causing, things that we can't foresee at this point. Now, the original rules for recombinant DNA were originally issued in 1976. relatively recently in kind of the history of humanity. But if we take a look at it, some of the rules in place were that there needed to be physical containment, so that we needed to have a location where we can work with these organisms, but it's going to be very unlikely or hopefully impossible for them to escape in the wild. And so you're going to minimize the risk of these kind of modified organisms, these changed organisms, from getting out of the laboratory and out into the environment. So there was physical containment, you had to be restricted, confined access and all that. But there was also a movement to have biological containment. This basically means that you're going to be modifying the organisms in some way that they're not going to survive outside of the laboratory. And this is, a good approach, but when I see this, I always think about the book by Crichton, Jurassic Park, which was made into a movie, where they basically, originally the original Jurassic Park movie, which is probably 25 or 30 years old now, they talk about the fact that they kind of engineered the dinosaurs so that they were dependent upon an amino acid being provided to them within their diet. And again, kind of related to those essential amino acids that we talked about before. But not to spoil the movie, but it's been out for 30 years, 25 or 30 years. So the dinosaurs kind of escaped from that controlled environment. They're able to reproduce on their own, and they're not dependent upon the food. So even though they were modified so that they couldn't survive outside of the laboratory setting, they adapted and they developed a way. And so there's a fear that, even with this biological containment, these modified organisms, aren't as vulnerable or susceptible as we think they might be. Now, the Recombinant DNA Advisory Committee, or RAC, continues to review and provide guidelines of potentially dangerous work. Most recently, they've been working on things like the drug-resistant microorganisms. In lecture, I think 2 lectures ago, we talked about MRSA, the antibiotic-resistant bacteria. And so there's a fear, again, that if you're working with these antibiotic-resistant bacteria, you're trying to study them, it's important if we want to try to figure out a better way to kill them off if they're disease-causing. If these things escape from the laboratory, they have the risk of causing massive disease outbreaks as they go through. If we're going to study genes for very highly toxic proteins, like the botulinum protein that causes botulism, it has an important use associated with it because we need to understand it to be able to treat it. But again, it has a huge risk factor associated with it because it could, potentially poison lots and lots of people if it was to escape. Organisms for which there's no current cure. If we're studying Ebola, it's a disease process, the virus in essence, that causes hemorrhagic fever. And it's primarily within the equatorial region of Africa. But if that was to get out and spread into the population, it could be devastating. So they're providing rules on that. And they're also taking a look at human gene transfer, which is going to be important next week when we start talking about gene therapy and the methods associated with kind of correcting or making kind of changes to the human genome in a way to hopefully correct a disease process. Now, the Recombinant DNA Advisory Committee, the RAC, that we talked about on these last couple of slides, remains active in making these recommendations. And so they've changed over time, over the, from 1976 to now, we're talking about maybe 45 years, but they're still providing guidelines on both basic science and clinical research involving recombinant DNA technology. And it's going to be this committee, among others, that's involved with gene therapy, looking at human gene transfer research. And again, this is an emerging field that has a lot of potential, but there are some risks associated with it. So we need to be able to understand what those risks are. Now, the whole idea of a transgenic organism, like the recombinant DNA that we've talked about before, is that we're going to implant in a new piece of DNA into a cell and hopefully have it incorporated into all of the cells within that new organism. And again, we're not talking about bacteria or yeast now at this point, single-celled organisms primarily. We're talking about multicellular organisms like plants and animals. And so what we're representing here is going to be a mouse model, a transgenic mouse model. And so again, to take a look at this, we're going to use some type of vector, and the vector is going to be kind of a mechanism to transfer our piece of DNA into the cells that we're interested in. And so, we talked about the bacterial plasmid, we were working with like bacteria, but when we start to work with other organisms, the plasmid's not going to work. And so generally what we're looking at with both plants and animals is that we're going to use a vector that's like a variation of a virus, so that, viruses are very good at infecting cells and injecting in an hereditable material and taking over the cells and getting their nucleic acids to take over and redirect the activity of the cells. But there are new ways to do that. There's nuclear injection, which is basically kind of shooting the DNA into the cells, into the nucleus, almost as if you're shooting it or injecting it in. And then in the last lecture, we talked about CRISPR, again, to go in and directly change the DNA of a cell. And so what we're looking at in order to create a transgenic organism is this idea that we want it to get into all of the cells within the organism, but most importantly, we want these new genes to get into the cells. They're going to be producing the eggs and the sperm. And so what we need to do is do the equivalent of in vitro fertilization. We're doing it with mouse embryos. We take mouse eggs and mouse sperm. We're going to do in vitro fertilization within a laboratory setting. So we're going to combine the mouse egg and the mouse sperm together. They're going to start to divide. So we get a fertilized egg. They're going to start to divide pretty much like what we talked about with in vitro fertilization previously. And what we're going to be looking at is that we're going to take our human DNA or our transgene DNA, we're going to inject it into these embryos. And so some of these embryos are going to have it incorporated by all of the cells. Some of them are not going to be incorporated at all. These are the solid blues. And some of them are going to be a little bit of a chimera. Some of the cells are going to have our new DNA and some of the cells are not. So we're going to have some blue and some red. So we're going to have these embryos that have been modified in some way and hopefully modified with our genes that we're interested in. So the red ones that we're looking at. And then these modified embryos are going to be implanted into a surrogate mother, just like in vitro fertilization, you allow the embryo to grow to a certain stage, and then you implant it into the uterus, and then that grows into an offspring. It's born, and then you can test it, and you test whether or not it's expressing that new transgene, that DNA that we inserted in. If we do this and it works out the way that we want it to, what we're going to end up with is this transgenic line of mice or transgenic line of organisms that are expressing this transgene, this recombinant gene that we inserted in, so that all of the cells in the organism have it. And most importantly, the cells that produce the eggs and sperm have it. And so this is going to be passed down from parent to offspring. And so we have this established line of these transgenic organisms. So the ways that we can do this, again, a viral vector is one way. And so a normal virus, like an HIV virus here, is going to infect a cell. When it infects its cell, it's going to inject in its viral RNA or its viral DNA. HIV uses RNA, but there's some DNA viruses as well. They're going to put that nucleic acid sequence into the cell that it infects, and that DNA ultimately, and RNA has to go through a step to, without worrying about the science associated with it, a step to get it into the DNA. That DNA kind of gets into and takes over the nucleus of the cell, so that this infected cell then is going to produce lots and lots of copies of the virus. So this is good if we want to infect a cell. If we want to, cause a disease process. But we don't want to do that. What we want to do is use this viral vector to insert the DNA that we're interested in into cells. And so what we're going to be looking at is using modified viral vectors. They're going to be modified so they can't replicate on their own. So they, again, like those guidelines that we talked about, they shouldn't be able to escape and live on their own. Jurassic Park says they might be able to. That's something else for science fiction or ethicists to kind of worry about. But basically, in theory, what's going to happen is that we get this viral particle. It still kind of infects the cell, but it's not capable of spreading. So it infects the cell, injects in the nucleic material, and incorporates that into the nucleus of the cell. but it doesn't produce more and more of this viral particles. And so basically what we end up with is this cell that has been modified. It's been genetically modified, recombinant DNA technology going in. So we've got that new DNA, this kind of light blue DNA within the darker black DNA within this nucleus. And so we've created then this recombinant organism. We've created this cell, which has a transgenic characteristic. We transfer the genes into the cell. It's been incorporated into the nucleus of the cell. Another way to do that, primarily within plants, but there is some work within humans, is to take our DNA, put it onto gold pellets, and then use what's referred to as a gene gun. But it's basically kind of this device that uses a lot of pressure and shoots these particles into the organism. So you can shoot it into an intact organism to the skin. And what you can do then is transfer the DNA into the cells in that way. And so we've often used this in plants. More recently, it's been used in certain types of DNA vaccines. So you use this and it basically shoots the DNA into the cells. It's incorporated by the cells and those cells start expressing whatever that DNA is. Another method to do this is to actually go in and do nuclear injection of the DNA. And so basically what you're doing here is collecting the eggs that we're going to be working with, in essence, collecting cells using a microinjector. And so we're basically using a very fine needle, which is going to allow us to poke a hole into a single cell, not into into the skin of the arm, but into a single cell. And we're going to inject that DNA in there in the hopes that the DNA that we're injecting into the cell is going to be incorporated into the nucleus, incorporated into the chromosome, into the DNA of the cell. And then it could be, passed on to the offspring. And we end up with kind of this transgenic organism here. So you kind of start out with a normal zebrafish. It's kind of a small fish that you can find in like aquarium stores. We inject it into the eggs, and then over time, you can see this kind of greenish tint to the zebrafish here, and it's that greenish tint that is expressing that new gene that we're interested in. So we created this new breed of zebrafish based on injecting in this foreign DNA. Now, an example of this is the glowfish. Again, you go into a pet store and you can find glowfish, trademark, fluorescent fish that you can buy. And so these are zebrafish, but you take a look at them. They were originally designed to do scientific experiments on polluted water because the color of the fish was going to change based on the amount of pollutant that's going to be present. But you can now buy these in most pet stores. The Food and Drug Administration basically looked at it and said, very seldom are people going to use this as a food source, so we don't have any guidance on this, and so we don't have to worry about that. And they basically said that these genetically modified glowfish, these fish that are modified, kind of similar to that tobacco plant with the luciferase gene in it. Now we have fluorescent zebrafish. No greater risk to the environment than normal zebrafish. Both of them are potentially going to be an invasive species. We let them out into the wild, but they're pretty to look at. They've got this interesting characteristic we can look at. Still, they're illegal in California, Canada, the European Union, and Australia. Again, because there is a fear that there's something going on with these fluorescent fish that we don't know about, and there are risks that we need to think about. Now, the whole idea, if we go in and try to modify the gene, potentially to fix a gene, is that we want to try to make sure that we don't cause more problems by going in and trying to fix them. So, we're going to put in our DNA into a cell, hopefully fixing one problem, but not causing a new one. And so because of that, we're going to do this lots and lots of times, ultimately ending up with a favorable mutation down here in a line that we're going to continue. So if we look at this diagram, what we're going to see is that we put it in here, and then sometimes it's going to be unfavorable. Sometimes it's going to be lethal, sometimes it's going to cause a minor effect, but a lot of times when we do this, it's going to disrupt the cell and kind of kill it off. And so we're going to do it multiple times, and then once we get a stable kind of organism here that has the DNA kind of incorporated into all of its cells, incorporated into the cells that are going to produce the eggs and the sperm, then we have something then that we can use as a transgenic line. So this is a pretty good overview. Come back for part 2, where we're going to look at some specific examples and concerns associated with these genetically modified organisms. And again, we're focusing in on higher organisms like plants and animals. As always, if you have any questions, feel free to email me. Thank you and have a great day.